that the processes of reproduction give rise to new individuals. But I'm sure you must have always wondered what determines the sex of the newly born individual, isn't it? Don't you ever think who, or rather what, decides whether the newborn is going to be a boy or a girl? Let us find out the answer to this. In different species, there are different factors that affect the sex of the newborn. In some animals, the temperature at which the fertilized eggs are kept is the deciding factor. Let us take some examples to understand this fact. In members of the order Testudines, for example, in turtle and tortoise, a temperature above 33 degrees Celsius leads to a female progeny, while temperature below 28 degrees Celsius leads to the birth of a male progeny. However, in lizards, a high temperature favors the birth of a male, while a temperature ranging below 28 degrees Celsius favors birth of a female progeny. There are also certain animals in which sex is not genetically determined. Snail is an example of such category of animals, as they can change sex as and when required. Sex determination. In human beings, the sex of an individual is largely genetically determined and depends entirely on the genes that we inherit from our parents. In diploid organisms having separate sexes, there are 22 pairs of chromosomes that are perfect pairs. They are called autosomes, whereas a specific pair which is not perfectly paired determines the sex of an individual. This pair is called the sex chromosomes. A female individual possesses a perfect pair of sex chromosomes having two similar X chromosomes, that is XX. These are homomorphic as they both are of same size and shape. A female, therefore, produces the same type of gametes called the ova. On the other hand, a male individual possesses one X and one Y chromosome, that is XY. These are heteromorphic since they differ in size. This is the reason why a male produces two different kinds of gametes, or the sperms, with half of them possessing X chromosome and the other half carrying Y chromosome. The sex of the child is determined at the time of fertilization, when the male and female gametes fuse to form a zygote. If a sperm carrying X chromosome fertilizes an egg carrying X chromosome, then the offspring will be a girl. And the resultant offspring will have XX combination of chromosomes. And fertilization of an egg by a sperm carrying Y chromosome produces a male offspring possessing XY combination of sex chromosomes. Thus, the sex of a child is largely determined by what they inherit from their father. In other words, it is the type of sperm fusing with the ovum at the time of fertilization which determines the sex of a child. There is therefore a 50% chance of the child being a male and a 50% chance of the child being a female. The same mechanism of sex determination is observed in Drosophila melanogaster, commonly known as fruit fly. Evolution Part 1. Extremely large number and types of living organisms are present on this earth, and interestingly, majority of them have not even been identified. Now the question arises, 1. How and from where did such a wide variety of organisms come to existence on this earth? 2. How did the human beings evolve? The constant process of gradual change occurring in the organisms since the origin of life that has given rise to enormous variety of complex organisms on this earth is called evolution. The concept of organic evolution was first given by Charles Darwin. Now let us discuss how the evolution occurred. During reproduction, variations arise either because of errors in DNA copying or as a result of sexual reproduction. Let us understand the process of evolution with the help of few examples. Let us imagine that a group of 12 red beetles live on green leaves of some bushes. These beetles have the natural tendency to increase in numbers by sexual reproduction and consequently generate variations. Let us also consider that crows are the natural predators of these beetles. The crows regulate the population of beetles by eating them, and therefore fewer beetles are available for sexual reproduction. Now let us assume that beetles develop color variation during reproduction. Consequently, one beetle develops green body color instead of normal red color. This beetle can inherit this variation in color onto its progeny so that all its offsprings are green in color. As a result, the crows cannot locate green-colored beetles on the green leaves of bushes, but continue to eat red beetles which are easily located on green leaves by them. As a result, number of green beetles in beetle population increases in each generation. In this situation, natural selection is exerted by the crows. More the crows are there, more red-colored beetles would be eaten up, and more will be the proportion of green beetles in the population. In other words, natural selection is directing the evolution in beetle population. Natural selection is the phenomenon wherein nature, in the wild, selects traits favorable to the species in its environment. Evolution Part 2. Another color variation arises in beetles during sexual reproduction, and in the progeny, one beetle develops blue body color instead of normal red. This beetle can also pass this color variation to its progeny so that all its offsprings are blue-colored beetles. Crows can detect both red and blue-colored beetles easily on green leaves of bushes and eat them regularly. As the population of the beetles expands, initially there are a few blue-colored beetles among the majority of red-colored beetles. At this point, an elephant comes by and stamps on the bushes where the beetles live. Consequently, most of the beetles get killed. By chance, most of the survived beetles are now blue-colored. This population again slowly expands and will contain mostly blue-colored beetles over a period of time. Here, color variation in beetles gave no survival advantage to them in the existing environment, as crows could easily detect and eat both red and blue beetles on green leaves. However, a simple incident, stamping by an elephant foot, killed majority of the red-colored beetles from the population. Survival of more blue beetles in the population changed the color characteristic from normal red to blue over a period of time. Had the red beetle population been very large, the accident would not have caused such a major havoc. We can say that in small populations, accidents can change the frequency of some genes in a population, even if these give no survival advantage to the possessors. The elimination of the genes of certain traits from the small population, when a section of the species population dies of natural calamity or migrates to other regions, is called genetic drift. It alters the gene frequency of the remaining population. Evolution, Part 3. As the beetle population starts expanding by sexual reproduction, a plant disease spreads, resulting in decrease in the amount of leaf material in the bushes. Expanding population of beetles is now poorly nourished. Consequently, the average weight of adult beetles decreases compared to what it used to be when the leaves in the bushes were in plenty. After a few years' time and a few beetle generations of such scarcity, the plant disease is eliminated. Now, more leaves of bushes are available as food for the beetles, and again, the beetles would gain weight. While considering this situation, we find that there are two kinds of traits in every organism. Inherited traits. These traits that are controlled by specific genes and are passed on from one generation to another. Any alteration in the DNA will be passed on through germ cells to the progeny, resulting in variations in them. Acquired traits. Certain traits are acquired by organisms in their lifetime. For instance, decrease in the body weight of beetles due to starvation is an acquired trait by the beetles during their lifetime. It involves changes in the non-reproductive tissues caused by environmental factors. Will not bring any change in the DNA. Therefore, even if some of the generations of beetles are low in weight because of starvation, this trait cannot be inherited by the progeny over generations. So we see that variation occurs because of sexual reproduction, natural selection, genetic drift. Evolution and classification. A plethora of organisms are present on Earth. Diversity in life forms is a measure of health of ecosystems. Different life forms may possess certain similarities among themselves. The existence of similarities among organisms allows us to group them together, thus enabling their study as a group. The placement of organisms into groups and subgroups on the basis of similarities and dissimilarities between them reveals their evolutionary relationship. The similarities or differences in the various characteristics form the basis for classification of organisms. But what actually are the characteristics of an organism? The details of appearance or behavior of an organism that distinguishes it from other organisms are called characteristics of that organism. The characteristics may be ancestral or basic characteristics and derived characteristics. The ancestral or basic characteristics are those that are shared by most members of a population and thus form their basic body design. On the other hand, the derived characteristics develop due to evolutionary changes in the ancestral characteristic. The evolutionary changes or derived characteristics lead to further classification of organisms into smaller subgroups. By identifying the hierarchies of characteristics between species, the evolutionary relationships of the species around us can be worked out. Evolution and classification. Basic characteristics. There are some basic characteristics that are shared by most organisms. For example, cells are the basic unit of life in all organisms. But we can classify different organisms based on the basic characteristics of the cells. Cells can be classified based on the nucleus. On the basis of this, organisms are broadly classified into two groups, namely eukaryotes, which are the organisms with a well-defined nucleus, and prokaryotes, which do not possess a well-defined nucleus. The eukaryotes can be further classified into unicellular organisms and multicellular organisms. With the specialization of cell types and tissues, organisms can be further classified on the basis of other basic characteristics. Their classification into plants and animals is based on their ability to perform photosynthesis. Furthermore, the non-photosynthetic multicellular organisms can be classified on the basis of whether the skeleton is present inside or around the body. Division on basis of level of body organization. On the basis of the level of body organization, the multicellular organisms can be further grouped into cellular level of organization as observed in sponges, tissue level of organization as observed in coelenterate, and organ system of level of organization as found in platyhelminthes. This gradual change from a simple to complex level of body organization is evolution. The presence of more number of common characteristics suggests closeness of the species. Also, the more closely related species are supposed to have had a common ancestor in the more recent past. Let us take an example to understand this better. Here, the boy and the girl are brother and sister, and so are closely related. This is because they have common ancestors in the first generation before them, that is, their parents. On the other hand, this girl and her first cousin are a little less related than the girl and her brother, because though the cousins do have common ancestors as their grandparents, they are in the second generation before them. Therefore, we can say that classification of species is in fact a reflection of their evolutionary relationship. We can thus form small groups of species with recent common ancestors, then supergroups of these groups with more distant common ancestors, and so on. Theoretically, moving backwards will take us to a single species at the very beginning of the evolutionary time. Let us now see if it is possible to trace evolutionary relationships. There are many evidences that help us in tracing evolutionary relationships. They can be categorized as morphological and anatomical evidences, embryological evidences, and paleontological evidences. Morphological and anatomical evidences. These evidences are based on the similarities in the external and internal features of the different organs in organisms. Based on this, they are categorized as homologous organs, analogous organs, and vestigial organs. Homologous organs. There are a number of organs in different groups of plants and animals which have similar basic structure and embryonic origin but are used for different functions. For example, forelimbs of a frog, a lizard, a bird, and human being show similarity in basic structure and development but are used to perform different functions. The forelimbs of a frog help the animal to prop up the front end of the body at rest and act as a shock absorber. The forelimbs of a lizard are modified for creeping movement. The forelimbs of birds are modified for flying, while the forelimbs of a man are used for holding, eating, writing, etc. The basic similarity in the forelimbs of these different vertebrates indicates that these vertebrates had a common ancestor who had 5-digited or pentadactyl limbs which got modified according to the special needs of the subsequent generations during the course of evolution. Analogous organs. Analogous organs are the ones which are quite different in fundamental structure and embryonic origin but perform same function and may even superficially look alike in entirely different species. For example, the wings of a butterfly and a bird. The wings of a butterfly are folds of membrane supported with a few muscles, while the bird's wings are formed of limb bones covered with flesh, skin and feathers. The insects and birds, in fact, have evolved from separate ancestors but use their wings to perform the same function, that is, flying. A similar example of analogous organs is that of the wings of a bat and a bird. Vestigial organs. The organs which occur in reduced or redundant form and are useless to the possessor but are homologous to the fully developed functional organ in the ancestors or the related forms are called vestigial organs. Few examples of vestigial organs are the vermiform appendix of the large intestine and the nictitating membrane in the human eye. Embryological and paleontological evidence. Let us now discuss other evidences to trace the evolutionary relationship. Embryological evidences. The similarity among the early embryos of vertebrates show that they have common ancestors. Paleontological evidences. Paleontology is the study of past life based on the fossil records. The study of fossils also helps us in tracing evolutionary relationships. Fossils are remains or impressions of the dead animals and plants that lived in the remote past. For example, the fossil bird Archaeopteryx lived in the Jurassic period about 180 million years ago. It had feathers, fused bones, and a beak. Like reptiles, they had their teeth in jaws, claws, forefingers, and a long tail. This suggests that birds have evolved from reptiles.